craniofacial biology also. So why it is widely used is because of its close genetic and developmental parallel with the humans. So, and what is the key advantages of using this model is because of the high genomic homology with the humans, which is nearly approximately 85%. And availability of numerous craniofacial specific mutant lines are present in the mouse. And well-established developmental staging, which is the Taylor stages, can be easily understood in this model, which is similar to the humans. And what are the tools which are used for the gene editing and the mutation to be caused is the Craniofacial, CRISPR-Cas9, transgenic importers, and the knock-ins and knockout methods. So in craniofacial application, what can be studied using this model would be the neural crest migration, palatogenesis and jaw formations can be studied, and mid-facial fusion and cranial suture development. So in these areas, what particular genes are studied so far is the MSX1, which is the knockout of this gene results in the cleft palate, and FGFR2 mutation model, which is the Apert and Trojan syndromes have been studied. So because mutation in FGFR2 results in the Apert and Trojan syndrome, and TCOF1 model results in the Treacher-Collins syndrome, and EYA161 involved in the craniofacial syndrome DOR, which is the bronchiotorhinal syndrome. So these 6 syndromes are actually studied in this model in craniofacial research. And this is the disease models which are studied is the cleft lip, cleft palate, craniosynostosis, micrognathia, and mandibulofacial dysostosis. So these are the key points which we have to remember, why the mouse model is used for the craniofacial development, and what are the genes studied so far, and what mutation causes what particular syndrome. So before moving on to the next animal model, there is actually a classification of animal model, like how, why these models have been used. There is a experimental model, which is experimentally, experimental reproduction condition mimics the human disease. So that is the experimental model. Next we have a negative model, where animal species in which a particular disease cannot be produced. Next we have the spontaneous model, where animal species has a disease that occurs naturally and mimics a human disease. Next is the orphan model, where animal species that does not mimic the human disease. So they are put under these 4 classifications. So whenever we study, we either take the experimental model, or a negative model, or a spontaneous model, or a orphan model. There's some other classification where we have this healthy animals, there occurs the spontaneous changes, genetically modified animals, which is the where we induce a particular disease to study our region of interest. So we can see the chemical or pharmacological induction, which results in cell transplantation or surgery, and after which we will identify the treatment of that particular disease. So next we have the most common animal model, which is the zebrafish, Dania vireo. So zebrafish offers a transparent and fast developing model system, which is ideal for live imaging and high throughput screening of craniofacial morphogenesis. So what are the key advantages of choosing this zebrafish is that there is optical transparency of embryos for imaging, high fecundity, and for statistical power, and external fertilization enables real-time observation. We can actually see how the embryo grows because it is actually very transparent. I have shown the images of the zebrafish in my earlier seminar, where we dealt only about the zebrafish exclusively. So tools for this is the same as the CRISPR-Cas9, morpholino antisense oligos, and transgenics. Similarly, the craniofacial application also includes the neural crest cell migration, which is being tracked via the GFP reporters, and viscerocranium and neurocranium development from neural crest. Jaw and pharyngeal arch patterning can be seen very clearly in the zebrafish model. So what are the relevant genes which have been studied so far is the SOX9A, SOX10, critical for the neural crest survival and cartilage differentiation, EDN1 required for the lower jaw formation, and BMP4 which influences the dorsoventral identity of the pharyngeal arches. So what are the disease models which have been developed as a craniofacial disc morphologies, frontonasal dysplasia, orofacial clefts, and ciliopathy, which is the orofacial digital syndrome. So next model we have the chick, that is the Gallus gallus. So why chicks? Because in class classic models for the craniofacial development used extensively for the tissue grafting and in vivo lineage tracing. So particularly we use chick for the tissue grafting purpose. So why we use chick is because they are large and easily accessible. So you can get avail it easily, and embryos develop ex utero also. So compatible with the quail chick chimeras for fate mapping. So tools like RCAS virus, in ovo electroporation and microsurgical manipulations can be easily done in the chick embryos. So regarding the craniofacial application, similarly NCC migration and patterning can be studied. Morphogen gradients in mid-phase development, palatal shelf elevation and fusion. You can either opt, you can go for zebrafish model, or you can choose stick whichever is easily accessible in your region or for your study. So for this, what are the genes which have been studied so far is the SHH, sonic hedgehog, which establishes the midline and facial quill, BMP4 and FGF8 involved with the facial patterning and proliferation, and the DLX gene family which controls the upper versus lower jaw identity. Just you have to remember any three genes which are commonly involved, so that if you will be able to answer whenever the questions are being asked regarding this. So the disease models also holoprosencephaly with SHH disruption, mid-facial clustering and facial asymmetry disorders can be studied in chick model. So next model will be the frog, Xenopus laevis, which is actually a powerful, frogs actually offers a powerful platform for early craniofacial gene function studies due to their large, easily manipulated embryos, similar to the chick which we saw. So advantage of this would be external fertilization and rapid development, ability to perform targeted injections and into blastomere stage, and tools is morpholinos, similar to the zebrafish model, mRNA injections, CRISPR-Cas9, especially in X tropicalis. Similarly, in craniofacial, you can use any of these models to study the NCC migrations, cartilage and bone specification in the head. So particularly we see the cartilage formation and bone formation in the head of the frog. So 3D imaging of facial development using confocal microscopy can also be done in the frog model. So far the relevant genes which are studied using the frog model is a twist, snail 1, 2, and which is used for the EMT and neural crest migration. So for EMT we actually go for the frog model, PITX2 left-right patterning of the craniofacial structures, and WNT and DMT pathways commonly involved in the facial mesenchyme induction. So next would be the fruit fly, Drosophila melanogaster. So despite lacking a vertebrate style face, Drosophila are indispensable for discovering the genetic networks and signaling pathways conserved in the craniofacial development. So why we choose this fruit fly is they have a very short life cycle and ease of genetic manipulation. So their life cycle is very short, and the tools which are used is a GAL4 UAS system, RNAi P element transgenics. So homologs of any craniofacial development genes can be studied in using this fruit fly. So it actually provides fundamental insights into the segmentation and patterning, organizing centers for the head development. So if you're interested in these areas, you go for this particular animal model. So relevant genes which are studied are the hedgehog, wingless and distal-less and engraved pax genes. So each of these have a specific role in limb patterning, mouth part formation, segment polarity, and determining the fate of those organ formation. So the last, we have two more animal models. So first would be the pig. Next would be the pig. So pigs are excellent translational models for human craniofacial surgeries and tissue engineering due to the anatomical similarity. So key advantage is because they have the similar size and morphology of the skull and palate in comparison to the humans. So comparable bone regeneration response will be there, and good model for dental and maxillofacial surgeries. So testing cleft lip and cleft palate surgical techniques will be one of the major craniofacial applications of using the pig model, and dental implant integration and bone grafting can also be studied, and distraction osteogenesis studies can also be done using the pig model. And the studies on biomaterials for alveolar cleft repair, palatal development and healing after surgery, imaging and biomechanical modeling are the recent researches which are done using the pig model. Next is the human pluripotent stem cells and organoid models. So patient-derived induced pluripotent stem cells and craniofacial organoids are emerging tools for studying the human-specific craniofacial disorders in vitro. So direct study of human disease phenotypes can be done, personalized genetic background can be understood, and 3D models can actually replicate the facial tissue architecture also. So differentiation of pluripotent stem cells into the neural crest cells, palatal fusion modeling using organoids, and gene editing for the disease modeling are the applications of using the pluripotent stem cells. So what are the relevant genes in this area is the CHD7, the charge syndrome modeling, PAX9 and IRF6, orofacial cleft studies, and TCOF1 mutations already mentioned involved in the Treacher-Collins syndrome. So yeah, all these animal models are the basically used animal models. So you have to know why they are used and why what are the anatomical similarities, and genomically also they will be similar to the human model. So for doing any research, we have only all these animal models for ease of use and access.